introduce Paul, who of course everybody knows, one of our current fellows. Paul did his undergraduate at Yale, and then moved on to Cleveland Clinic, the Lerner School of Medicine, where he did his MD, and then did his residency at University of Pennsylvania. He's been with us for 6 months now, and he's going to talk to us about traumatic brain injury work he's been doing.
All right, thank you. Good morning. Of course, I want to thank everyone for having me here this year, not just here this morning to tell you about some of the work that I've been doing. It's so far been a great learning experience, and I hope you find some of what I talked about this morning interesting. So I'm going to be giving you basically a taste of some of the work that I had been doing at Penn, largely as a resident in a systems neuroscience laboratory that is interested in how networks, specifically hippocampal networks, become dysfunctional after traumatic brain injury, and then hopefully talk about some of the future directions from there. So I will not belabor this because we always do when we give an intro, but I do think it is worth mentioning that TBI really is a big problem, as we all know, especially working at an institution like Grady. 50 million a year in the world, huge costs to the economy, both direct and indirect, and not just the economic costs, but the chronic disabilities associated with traumatic brain injury are huge, and they run the gamut from cognitive problems, behavioral problems, movement disorders, epilepsy, etc. So because of these things, some consider one of the more complex diseases of this central nervous system, and one that we know very, very little about, despite decades of research. So I thought this quote was interesting. The most recent major breakthrough in clinical management of TBI was the introduction of computed tomography scanning in routine care about 40 years ago. Since then, there have been no major improvements in outcome after TBI. And this was written just 2 months ago, a little less. So I'm going to talk a little bit. I'm going to now drill way down, talk about some of the work that I've done in memory impairment after TBI, and in the context, or as thinking about it as a failure of hippocampal networks to communicate. And this is largely, we'll go over some studies in rodents. Then I'll touch briefly going up the phylogenetic tree to pigs, which are a gyroencephalic species and have a much closer gray to white matter ratio that to humans than a rat does. And I'll touch briefly on the work that we did trying to develop a way to implant an awake pig with an electrode in the hippocampus and try to glean some information there. And then I'll go over a couple of future directions. Cognitive deficits post-TBI. So they are significant. We've known for a long time that chronic memory and other cognitive problems are a significant problem in moderate and severe TBI. You can see a well-known study on the bottom left there showing that these things persist on a broad range of cognitive performance. But more recently, we've also known that mild TBI can cause significant impairment. And specifically, hippocampal-dependent memory has been shown to be persistently problematic after even mild TBI. And there was also a study in 2008 looking at people coming back from the Iraq and Afghanistan wars, and at least a quarter of them were having persistent memory problems, among other things. So one way to look at this is through animal models of TBI, and to what degree animal models translate into the human condition is the fundamental problem in TBI research. But nonetheless, there are a couple of well-established models that can say something about processes that may be initiated by an injury and the consequences thereof. So the one that I'm going to talk about this morning is the lateral fluid percussion injury, which is a well-established model, a mixed model of TBI in rodents, and in this case in rats. Basically, a small craniectomy is made over the left frontoparietal region between the between the coronal suture and the lambda suture. You hook up a closed column of fluid. You drop this piston, and it initiates a pressure wave that gets translated to right over that craniectomy, and it causes an instant deformation of the brain. It has many advantages. Some say it mimics, to some degree, the mixed nature of human TBI, because there's both a focal contusional component often, as well as a axonal injury that seems to be more diffuse. It's titratable, so because of the collective experience, we know that we can drop the pendulum from a higher distance, and we can get a reproducible, higher degree of pathology. And we also know that there are behavioral differences, including spatial memory, which are reliably deficient after this injury. So it is one of the most widely used models of TBI, and it's the one that we chose to look at. This is one of the original studies demonstrating that there is spatial memory impairment after this injury. This is using the Morris water maze. We won't go into too much detail, but basically there's a circular tub of water and a hidden platform that's submerged just beneath the surface. The rat's introduced into the bathtub, and he has to swim around and find this platform. You do this on many trials, and he learns where it is. And then in the case of investigating TBI, after injury, you can do a number of things, but the most well-established is to then put the rat who has already learned where the platform is into the water and see how long it takes him to find it and how long he spends various distances from the platform. And using this, you can get a reliable deficit in spatial working memory, shown on the right there. And indeed, the higher the injury level, the higher the spatial memory deficit. Same model. This is one of the original papers showing that there is indeed pathology in the hippocampus after FPI. What's interesting about this is for such a significant force applied to the brain, this is not actually that much pathology. And what we're seeing here is cellular dropout in the pyramidal cell layer in the CA2/CA3 region of the dorsal hippocampus of the rat. And this is at 1 week post-injury. This was done in the 90s, where most of the focus on TBI pathology was on neuronal injury. And indeed, there is neuronal injury in this model. But we know now that a major component of TBI is axonal injury. And studies to characterize what exactly the axonal injury are in this model and in other models are ongoing, and I'll show you a little bit of that later. So how do we think about this in the Wolf lab, which is where I was at Penn? We have an inciting injury. You had acute, ongoing injury mechanisms, maybe inflammation, you get some cell loss, axonal injury, maybe some synaptic changes. This results in the pathology that we can see and measure. And somehow this leads to memory impairment, and we kind of group that into some kind of failure of hippocampal network communication. But how do we begin to investigate what exactly that means? What are those mechanisms, and how, if we understand them better, can we potentially intervene upon them? So people have begun to do this kind of work, and one of the more natural things that we think about is, can we just place an electrode in the hippocampus, measure what the neurons are doing, and from that perspective, try to understand what these mechanisms of network miscommunication might be. So a few studies have been done on looking at theta oscillations in the hippocampus, or theta power in the hippocampus, and some indication that it might be reduced after fluid percussion injury. So hippocampal theta, we'll call it for now, is a particular band of frequency oscillation in the local field potential that is often measured in the hippocampus, primarily these days in rats, that seems to be associated with active moving and exploring, as well as REM sleep. It's an established correlate of memory performance in rats, and this is well established in the literature, that if you knock out theta power in the hippocampus, you reduce memory performance in rats. You can even restore theta to a limited capacity and get improvements in memory again in a limited capacity. We mentioned that power is high during exploratory activity. Performance goes down when we disrupt it, and we can rescue behavior to a limited degree. So there was a study in 2010. I think it was out of UC Davis or someplace in California. Hippocampal theta seems to be reduced during exploratory activity after TBI or after fluid percussion injury, but not during spatial memory performance. Then actually, this grand rounds here at Emory has become something of a series in this. We heard from someone a couple weeks ago who had conducted a couple of studies looking at this question of reduced hippocampal theta, and they found also that there was reduced theta power, and that in their studies it did correlate with spatial memory deficits. And then they went on to stimulate the medial septum, which has diffuse projections into the hippocampus at a theta frequency, and may have found some improvements in performance. This is an interesting study in light of those findings. So this group from Akiva Cohen at actually the Children's Hospital in Pennsylvania asked the question, well, is this drop in power in theta specific to the theta band, or are there broader deficits in power? And they found when they basically did a regression analysis of a certain type to correct for broadband power drops, they found indeed there was a significant drop across a wide range of frequencies. And once they did that, the residual differences didn't quite make significance in theta, throwing into question whether what we see sometimes when we measure a dropout in theta in the hippocampus is this specific to theta, or does this represent some more global process? For example, a decrease in multi-unit activity firing, or other widespread deficits in synaptic input. But
Do frequencies slower than theta tend to go up, like delta? The idea that you're just slowing everything down?
no, so delta's down as well. So I think the range is something from like 2 up to at least the sort of mid gamma range, 30 to 50, something like that is what they looked at. We can go back and look at it. No, they didn't really find any specific other frequency that popped out, at least in that study. Now, were they looking for that? I don't think so. But
These you're still all talking about rats.
this is all rats. This is all rats. But I think we need to go deeper. So when we say a theta oscillation recorded in the LFP, what does that really mean? What does that mean biologically, and what does that tell us about how a network is communicating? So now things will get a little bit more technical. So one place to start is to talk about how we measure network communication with extracellular recordings. So when you put an electrode near a group of neurons, you can record a raw signal, which you can see at the top there. And this is unfiltered. The range of frequencies can go from anywhere from like 0.5 or lower hertz up through like 9,000 hertz. And if you don't filter that and you look at the tracing, you might get something like what you see at the top. Now, if you filter the signal, higher frequencies tend to represent local action potential current fluctuations. So as a rough pass, we can say that higher frequencies from 600, say, to 6,000 hertz gives you information about local neurons that are spiking, and you can actually extract the timing of individual of neurons firing. You can extract those individual action potentials, and then you can actually sort them and make some assumptions about which ones are coming from what individual neurons. So now you can get spike trains from individual neurons and say that this neuron is firing in this pattern at this rate, this neuron's firing at this pattern, and so forth. If you filter the signal for lower signals, like say up to 150, 200 hertz, where things start to get a little bit murkier, but certainly in the lower frequencies, this LFP, we know, is largely dominated by synaptic input into that region. So when you see a theta oscillation, for example, or an alpha oscillation, or a delta oscillation in a particular region, you're not seeing how those neurons necessarily are firing at that level. You are seeing aggregate synchronized synaptic input into that region. And that's really important because now if we can measure both the action potential patterns and the input, we can say something about how that region is responding to its inputs and how at that node communication may be happening or not happening appropriately. So let's say something about, in that context, a theta oscillation and how it might, how we think in rodents at least, it might be organizing activities in the hippocampus. So one of the major findings in rodent hippocampal physiology is the idea of a place cell, which is a pyramidal cell that fires faster when a rat is in a particular location in its environment. So if it's running around in a box and one of its place cells fires in the upper right quadrant, it will fire more when the rat is there. It'll stop firing when he moves away. We know that these place cells and other types of pyramidal cells are entrained to theta input coming into the hippocampus. So on the bottom left there, you can see those neurons are firing at certain phases of the theta oscillation. And if you dig into this deeper, and this is work that's been done by Moser, by Bujaki over many decades, if you track individual place cells and where they code in a field, and you look at a path that a rat is traveling, there are individual place cells that will fire in sequence. We know that those place cells are entrained to theta at specific phases, and that those phases shift reliably as the rat moves. It's a function of position and a function of velocity. The point being that those oscillations are doing something very important to organize these place cells. But theta oscillations are not the only inputs into the hippocampus that are doing this. So there's something called a ripple oscillation. These are higher frequency in the range of 110 to 200 hertz, and they're short duration, about 100 milliseconds, 150 milliseconds. They also occur in the pyramidal cell layer, and they are in the LFP. If you record simultaneously from deeper down in the dendritic arbor, they seem to be associated with sharp waves or large currents of input from CA3, from the Schaefer collaterals. We know that these events tend not to occur when theta is dominant, but are in a different state. So they tend to occur when a rat is quietly but alertly resting, or it can occur during non-REM sleep. And it's thought that these ripples have something to do with memory consolidation, maybe memory distribution outside of or into a wider hippocampal network. And we know, and this is some of the more remarkable data that's come out of rodent physiology recently. So what you're seeing here, the colored little slashes are different place cells that are firing as a rat moves along a track. So he's moving in time from left to right, and there's sort of a plot of his velocity down on the bottom. And as he traverses that particular path, a very particular sequence of place cells is firing. And those we know are entrained to the theta oscillation, which is sort of represented at the top. Remarkably, people have found that just prior to this, before the rat is moving, that exact sequence of place cells is replayed before the animal actually walks, but it's sped up in time so that those place cells fire in the correct order, entrained to that ripple oscillation. And then even more remarkably, I think, after the animal has traversed that path, that sequence is replayed again, but in reverse order, again entrained to a ripple. This is some of the major data that's led people to think that this has some major role in how memory and memory engrams are formed and maybe distributed beyond the hippocampus. Ripples have been noted in other regions, such as the prefrontal cortex. So how does this relate potentially to TBI? Well, so I think this suggests that oscillatory inputs into the hippocampus are extremely important in organizing sequences or neuronal ensembles, and that this is one mechanism by which information is carried and manipulated. And we think that it has something to do with memory, certainly spatial memory, potentially other types of episodic memory. TBI, those oscillations, those inputs coming in may not be functioning properly, or they may not be able to properly organize those cells that are firing for any number of reasons, for any number of mechanisms. And this, of course, may lead to memory impairment. So our hypotheses: derangement in the synchronization within intrahippocampal connections may be a mechanism by which spatial memory impairment happens after TBI. This could be poor hippocampal oscillatory function, both in terms of its power, its ability to come in from an outside system, or its ability to organize neuronal ensembles locally. This could be an interneuronal problem locally. This could be an axonal injury problem leading to reduced inputs. And this could also be both intrahippocampally and extrahippocampally. So in light of this, one of the things that I did in the laboratory is undertook a smaller exploratory study asking, are there changes in the inputs into CA1, specifically into the dendritic arbor after fluid percussion injury? What is the nature of those changes? And secondly, are the firing patterns of CA1 neurons altered in response to those inputs? So briefly, the design: a small study, 20 rats, 10 of them underwent injury, 10 of them received a sham surgery. All of them, the day after injury, underwent Morris-Water maze testing. And then subsequently at day 7, 5 of them from each group underwent acute electrophysiology under anesthesia. And what I'll be talking about today are high-density recordings in the hippocampus during those recording sessions. But we also simultaneously measured spiking activity in the medial septum, and we also undertook some stimulation studies of the perforant pathway into the hippocampus. And again, today we're going to talk about the intrahippocampal findings. As you can see on the bottom left, our injury level, which may or may not mean anything, it was 2.01 to 2.46. This is on the moderate to severe level of injury for this model. This is just demonstrating that our data. Oh, sorry. Go ahead.
In terms of, you examined them under anesthesia. Was it, do you know what anesthesia is used?
Isoflurane.
You didn't find it, because in my own stuff with hippocampus, I found that a lot of times the primal layers are severely depressed by isoflurane in terms of just their activity level.
Yeah. So, and we can get into that. So, I think the idea of looking at theta oscillations, for example, becomes largely difficult under isoflurane anesthesia. And you're right. If you titrate up the isoflurane levels, you can severely depress spiking activity, for example, in the pyramidal cell layer. So we used a minimum amount of isoflurane, but it's important that you brought that up because the study was designed to undertake investigating the intrinsic networks within CA1 and how those connections may be altered. It's not asking questions about what is going on during specific behaviors. So we're still, go ahead.
Aren't those networks so much behaviorally dependent?
They are, but they still have a function under anesthesia. So maybe we can talk about it further as we go along here. So they all underwent Morris-Water maze, and this is simply demonstrating that they all indeed had a memory impairment after injury. And this is showing that we also saw on the left, you may not be able to make that out so well actually from out there, but we saw a dropout in the cellular layer in CA2 and CA3. And the slide on the right is just showing, and this is the characterization of this is currently ongoing, but this is APP pathology. So APP is a marker of acute axonal injury that is used to define axonal injury after TBI, among other things. It's a microtubule breakdown product, and this is simply demonstrating that we do have axonal injury, significantly so in this model. And this is at 7 days after injury. So what are we measuring? We have an electrode sitting in the dorsal hippocampus that is 32 channels. That's extending down the laminar structure of the hippocampus. And the organization of the hippocampus allows us because those inputs are arranged in an orderly way below the pyramidal cell layer, we can start to say something, depending on which electrode is showing us activity, what inputs or what may be playing a role in how the response is occurring in the pyramidal cell layer. So at the top, channels near the top would give us information about inputs near the pyramidal cell layer, but spiking activity about the outputs of the pyramidal cell, pyramidal cells. Whereas deeper down in the middle and towards the bottom of our probe, this is the stratum radiatum, as you'll see. And we know that the Schaefer collaterals come in there. That can tell us something about inputs from CA3. Perforant path inputs tend to come in deeper down, and those are from the entorhinal cortex. There we go. So this is what a raw tracing looks like from 1 second from a single sham rat. So this would be our uninjured rat. And you can see that the spiking activity riding on top of these lower frequency oscillations, and I have it lined up with our cartoon of a CA1 region on the right. And so you can see not only is there spiking activity at the pyramidal cell layer, but you can see a higher oscillation occurring right around here, which, because of the laminar arrangement of the hippocampus, suggests that this may be the stratum radiatum and therefore inputs from CA3. So just, again, it's important to look directly at your data. So if we look at 1 second from an injured rat, there we go, lined up in a similar fashion, we can start to see already that there may be differences. Specifically, or interestingly, this input in the stratum radiatum looks like it may be altered, attenuated, or changed in some way. So, can we quantify this? So if we find the channel on our electrode that has the maximal, we're actually looking at ripple frequency power as a localizer for our pyramidal cell layer. We know that this localizes from previous literature to the pyramidal cell layer. So if we define the channel of the pyramidal cell layer, we can then line up all of our recordings, and we can get aggregate measures of the of inputs coming in. So this is actually again an example of a single sham rat. The graph on the left is showing oscillatory power up through 50 hertz, and you can see this is that oscillation that we saw in the raw tracing. It's about a 10 to 20 hertz oscillation localized into the stratum radiatum. This, by the way, among other things, is delta frequencies, and there's some theta frequency in there as well, but it is definitely not dominant under anesthesia. These are higher frequencies, and this is showing that we have a bump in power in the it's about 110 to 250 range, and this, interestingly, is the ripple frequency range, but these are not necessarily ripples. So now, if we aggregate them all together, line them up, and then look at a comparison between sham and injured animals, a couple of things pop out. Two things pop out, actually. A dropout in the 10 to 20 hertz range down here, maximal at the bottom of the stratum radiatum, but extending up into it a little higher, and a dropout in our in this higher frequency range, about 175 to 300 or so. So this confirms what we saw in the raw data. You know, it's in this may represent a dropout in projections from CA3. This may represent power dropouts in intriguingly in the ripple frequency range. This could be a dropout in ripples. We don't know that. Of note, there's no difference in the theta frequency range, but again, this may not be very relevant under anesthesia. So the next question is, given that that may be loss of CA3 input, this may be loss of power in the ripple frequency range. We know that CA3 input drives ripples. You know, is there a relationship between those two regions? So looking again at the raw tracing in a sham animal, the top tracing is filtered from 110 to 200, which is the ripple frequency range. And we can see that the power fluctuations or the amplitude fluctuations come in episodes reminiscent of ripples. And the bottom is that 10 to 20 hertz oscillation down in the stratum radiatum. If we impose an envelope on the amplitude fluctuations, we can just kind of see that there seems to be perhaps some coupling between the two, between the phase of the lower frequency in the stratum radiatum and the amplitude of the higher frequencies in this pyramidal cell layer.
What's the frequency?
Which one?
If you're slower and you're faster.
The slower is 10 to 20 hertz, and the faster is, on this graph, is 110 to 200.
Not directly analogous to a theta gamma.
It's not exactly analogous to that, although it is a similar analysis. There are many ways that you can look at theta gamma, right? So phase amplitude coupling, which is what this is leading towards, you can do that also with theta gamma, but you can also do phase coupling with between theta and gamma, which I think is also looked at. If we look in the injured animals, it seems like that phase correlation with amplitude of the higher frequency may not be as preserved. Can we formalize this? So if we do a formal phase amplitude coupling analysis, we see that relationship between the stratum radiatum input and the pyramidal cell layer higher frequency oscillations is dropping out in the injured animals. This, interestingly, is the high gamma frequency, and there doesn't seem to be a difference there. The implications of that, I'm not sure yet. But we do seem to see a dropout in that coupling. So FPI demonstrates expected spatial memory deficits. There's a loss of input into the stratum radiatum of CA1 after FPI. This may be coming from CA3 projections, which we know has been injured in this model. Loss of high frequency power in the pyramidal cell layer after FPI may be a loss of ripple-like events. The amplitude modulation of these high frequency oscillations is driven in the normal rats by input into the stratum radiatum. This coupling is diminished after FPI. This could suggest that at 7 days after FPI, there is a loss of CA3 input into CA1, which has a diminished ability to drive higher frequency oscillations, important for spatial memory. This then leads us to how are the local neurons responding to this? How is their firing pattern altered in response to these changing inputs? So we figured that pyramidal cells and interneurons may be responding differently. So using an extracellular analysis technique that we borrowed from Brzezinski's group from the 90s, we were able to extract certain features about the wave forms and other features of spikes, and to sort them into putative pyramidal cells and putative interneurons. And this is just a three-dimensional graph showing how that divides out in our data. Doing this, on the left, we basically can talk about different cell types that we've recorded from. So we had 112 pyramidal cells that were isolated, and 112 total neurons that are about 65% pyramidal cells in the sham group, and then 83 neurons in the injured group, which also about 66% pyramidal neurons. So there didn't seem to be a major shift in ratio of pyramidal cells to interneurons. Within the interneuronal group, we've classified them by their location. So we know there are interneurons in the pyramidal cell layer. There are interneurons above and below. Those ratios shifted a little bit, but the numbers were fairly small. So Not sure what we can conclude from that. And so then the first thing to look at is how are those, is the firing rate of those neurons at all altered? If you have a loss of what seems to be in this state being under isoflurane anesthesia, a major input coming in, a regular input coming into the stratum radiatum, losing that input, does that alter the firing rates of these cells because they're losing synaptic input? And it turns out that no, these cells are still able to fire overall at the same rate. That's true for both the pyramidal cells and the interneurons. There's no change there.
How are you identifying these different cell populations?
So,
Based on waveform or pattern?
so it's a combination of three features.
Okay.
So we know that pyramidal cells tend to have a more bursty firing pattern. That's one. Two is their intrinsic firing rates in interneurons tend to fire faster than pyramidal cells. Three, the spike width, so the waveform shape, and using those you can classify them.
In this analysis,
Which one? This one?
the next one, next. So you record the same firing rates of cells you can record,
Right.
but
So-
you're not necessarily recording
That's right.
the unhealthy cells.
Correct. So they-
the volume or number of cells issue. Are you able to count that at all? Is it harder to find the cells of interest in your animal versus?
Yes, it absolutely is. And so in order to even count these cells, you need a certain minimum number of spikes over the course of your recording. So yes, this doesn't suggest that there are no cells that are injured or cells that are in the process of dying or have died in that area. That's absolutely true. But those that are functioning, so my cutoff was 100 spikes over the course of 15 minutes. So there may be cells that are firing less than that under anesthesia. But of those, and that's pretty slow. So cells that are faster than that, there's no change in their firing rate. But yes, absolutely, this doesn't suggest that there aren't injured cells there that are just not firing.
Because what I'm saying is maybe the deficits have more to do with the fact that you don't have enough of the cells doing this. You're still recording cells doing this.
It's possible.
But maybe there's only a tenth of them, 50 members.
Right, but there's no difference in the number of cells that we recorded. So in that scenario, you would have to posit that there are some cells that are sick, more so in the injured case, that don't meet that criteria that you can't record from, that may be doing things in other states, like not under anesthesia, that could be contributing to problems. But I think we didn't isolate a significantly different number of sufficiently fast spiking neurons in one group than the other.
Maybe 10% in the healthy group and 90% in the unhealthy group.
No.
You said it was more difficult to find the ones in the injured.
No. It wasn't actually. For a long time, before I knew any of these ones, I actually thought it was easier. I thought that we were getting more activity in the injured ones. But.
And is there any difference in the ratio of pyramidal cells to interneurons?
No. Oh, that's right. No. So it's about 65% pyramidal neurons in both cases. Sorry, it's taking a while to advance. All right. So if their overall firing rates seem to be unchanged, at least those neurons that we can record, what about their timing? So if we look back at a previous slide, you notice that these cells may be riding on top of these major inputs coming in. So we can look at that. We can ask that question. We can ask, are these cells firing entrained to that slower oscillation? And indeed, in the Sham case, 100% of the isolated pyramidal cells were entrained to that oscillation, the stratum radiatum. And importantly, this entrainment analysis was looking at the phase of that oscillation deep down in the dendritic layer, not that oscillation at the recording in the pyramidal cell layer, but literally where it's coming in the dendritic arbor, and looking to see what phase that neuron prefers to fire. And if we aggregate that input, aggregate that data, 100% of them are significantly entrained to that oscillation in the sham case. In the injured case, only 83% of those neurons had any form of entrainment to the phase of that oscillation. And further, those that were entrained in both groups, in the injured group, and then this is the graph on the bottom left, those that were entrained showed a reduction in the degree to which they were entrained. So on the y-axis there is a measure called the resultant vector length, which is basically a measure of variance in circular statistics, showing you how entrained. So a value of 1 would mean that every single neuron is or every single spike is exactly at the same phase, and a value of 0 would mean it is completely random. And that's for pyramidal cells. Interneurons were also highly entrained, but we didn't see a difference in the number of them entrained between sham and injured groups, but we did see a reduction in the degree to which they were entrained. Now, if we look at the preferred phase of each cell, so we know that the pyramidal cells are entrained to that oscillation, but what phase of that oscillation do they tend to be entrained to? That's the graph on the left there. They all seem to cluster fairly strikingly around a smaller range of phases just on the descending slope of the oscillation. And that's a pyramidal cell. If we look at in the injured guys, that tight distribution has gone away. There are some that are entrained at that phase, but there are many more that are actually entrained slightly later, and some that are entrained slightly earlier, and the spread is a bit wider. Interneurons also show a similar pattern. So sham neurons, interneurons, a bit wider of a distribution than the pyramidal cells, and this is probably reflects the fact that there are many different types of interneurons. This is grouping them from all the different layers. So given that, it's actually remarkable that we do see a phase preference for the interneurons as a group in the shams. That distribution becomes further dispersed in the interneurons. So we have a marked reduction in this 10 to 20 hertz input into the stratum radiatum. This was likely attenuated CA3 inputs that's 7 days later under anesthesia. We saw no change in theta oscillatory power, but this may not be relevant under anesthesia. This is accompanied by a power loss at higher frequency, about 175 to 250 in the pyramidal cell layer, and independent of that power reduction, a decoupling of that lower frequency input potentially from CA3 with a higher frequency oscillation. This could represent an attenuation of CA3 inputs, driving ripple-producing synaptic activity in CA1. However, despite this input power loss, both pyramidal cells and interneurons are able to maintain normal firing rates. Yet, both cell types demonstrate a significantly attenuated ability to maintain proper entrainment to that residual CA3 input. So it makes one wonder, how is this possible? How do you get a loss of input, but still a group of cells that are largely able to maintain a normal firing rate? So in the schematic, you have an inciting event or traumatic injury. You have the pathological processes that are going on. This may lead through a variety of mechanisms to loss of synaptic input into CA1, and then over some period of time, in the case of our study, potentially 7 days, the circuit has evolved. It's potentially compensated for that loss of input. Maybe this is why it's able to eventually maintain its firing rate. Has it increased its gain at a postsynaptic level? Has it retuned its neurons to maintain their firing rate in the face of reduced input? And does this, by doing this, the consequences of this loss of the correct timing of firing, potentially even a hyperactive response to normal inputs? And could these mechanisms be contributing to memory impairment? So this idea of hyperactive response to normal inputs, I want to touch on that. So concurrently with the rat work, there's the Wolf Lab makes use of a purely diffuse axonal injury model that's been developed at the University of Pennsylvania. This is a rotational injury model in pigs. So a pig is placed in this scary-looking device on the right called a hyge, where the head is rotated very quickly, and this induces a rotational injury and is largely purely axonal injury. So at the correct levels, there are no contusions, there's no hemorrhages, including petechial hemorrhages. There's purely axonal injury, and this is just a slide demonstrating at the top the APP stained axonal pathology in a pig after one of these injuries, and then by comparison, this is human pathology of a 20-year-old who had a TBI, and this is 48 hours after his injury obviously led to death, showing a similar axonal pathology.
You said in the pigs there was no petechial injury, no
Correct.
contusions, all that sort of stuff?
That's right.
So like a CT negative.
Correct.
Brain injury.
Right. And as you can imagine, at Penn, this model is being used to look at mild TBI and concussion and those sorts of things. So I just want to emphasize one aspect of one study that was done in those animals. So in this study, the pigs were injured, and then at 7 days they were sacrificed. The brains taken out, and slices through the hippocampus were done, and slice electrophysiology was performed on hippocampal slices. And what I want to emphasize is they had a stimulating electrode in CA3, that a recording electrode in CA1, and this is showing different input currents, stimulating currents. This is showing a fiber valley slope, which is basically the presynaptic input, the level of presynaptic input generated for each level of current stimulated. And what I wanted you to focus on is the green line, which is Sham's, showing the relationship between the amount of current and the amount of presynaptic input into CA1, and the blue line, which is the 7-day coronal plane injury, showing the same curve. And you can see that for a given amount of input from CA3, CA1 is seeing a markedly reduced amount of presynaptic activity. So the current being injected in CA3 results in reduced presynaptic activity at CA1. On the right, in light of this, now we've plotted the fiber volleys on the x-axis. So for a given presynaptic input into CA1, you get a certain amount of EPSP slope, which is basically a marker of postsynaptic response, population postsynaptic response. So for a given input, you have an exaggerated output. And the blue is the TBI group, the green is the Sham group. So injecting a small amount of current results in reduced presynaptic input seen in CA1, but that given amount of presynaptic input results in an exaggerated response by those CA1 neurons. And this is suggestive, again, of a hyperactive circuit that may be a result of compensatory mechanisms secondary to loss of input from TBI. So, really the major project that I was working on in the lab, in addition to this rat project, was developing a method of implanting electrodes into the hippocampus of awake pigs. Because you can imagine, as Neil pointed out astutely, you have a lot of issues under anesthesia with understanding the circuitry, and the relevance of any finding to what you really want to know is the behavior of the animal and how that's been affected by TBI and what the electrophysiological correlates are. So, and I'm not going to belabor this, but basically we developed a method. We developed a new stereotax that allowed us to use a relative coordinate system in order to implant an electrode into the hippocampus of a pig. We borrowed techniques from deep brain stimulation. We mapped in the sagittal plane the hippocampus going down into the thalamus, and then based on these maps, we would place our electrodes. We developed a method of securing down these electrodes and getting them to a state where they would withstand the robust activity of a pig. And I guess that video is not going to play, But now we have a high density probe sitting, again, analogous to the rat situation in the hippocampus, but this time the animal is awake and behaving, and we can look at different states. A couple of findings. This is, again, not an injury, but in normal animals. One of the questions is, does all of the physiology that we've seen in the rodent hippocampus have any relevance to gyrencephalic human brains? And this is some preliminary data suggesting that it may, to the extent that the pig brain is more analogous to the human brain than the rat is. So this is simply showing that there is a robust theta power signal in the hippocampus during movement, and that when the animal stops moving, that theta power drops out. Pigs also have ripples that are accompanied by inputs into the stratum radiatum. And pigs also have place cells. This was during a wake. It's wake activity. So yeah, you can record from them, and you can see ripples when during quiet periods of rest. We since then do have sleep data, I believe. I haven't seen it though, and this is simply showing there are place cells. All right, so so in summary, I want to say that a conceptualization of what's going on in TBI starts to emerge. Some inciting event happens, some period of time goes by where the circuitry may be altered or altering, and this could be made more complex by ongoing injury processes, but also by compensatory responses to changing inputs. And this leads to a compensated but abnormal circuitry, particularly under stress or under behavior. And this could lead to many of the disturbances that we see after TBI. So one area of interest is post-traumatic epilepsy. We know that maybe 20% of severe TBI patients develop PTE. 20% of symptomatic epilepsies result from TBI. This is maybe 4% by some counts of all focal epilepsies. 5% of patients referred to major epilepsy centers like Emory are thought to have TBI as a major risk factor and component of their epilepsy. And 35 to 62% of epilepsy due to trauma, regardless of where a focal injury may have been, is mesial temporal lobe epilepsy. So can we see this period of time after injury, the acute period of time, being days to weeks to months, depending on the type of injury, is this a period of epileptogenesis? And is this a period of time, an opportunity for investigating how TBI may lead to network changes that could result in PTE, cognitive deficits, and so forth? In the PTE world, we talk about early seizures, which are less than 7 days, and we think that that's largely due to the mechanisms of direct injury to the cortex, and late developing seizures, which evolves into post-traumatic epilepsy. And what is the relationship between the two? Some clinical data suggests that early seizures are a risk factor for post-traumatic epilepsy, although there is some controversy there. But we know very, very little about what those mechanisms might be, what the electrophysiological signatures may be in that acute phase that put a person at risk for developing PTE and other sequelae. So where do initial seizures begin? Does this begin in the perilesional cortex and tissue? Does this change over time? Are there certain electrographic events like epileptiform discharges, sharp waves, pathological high-frequency oscillations that can be predictive of developing late seizures? And what might their mechanisms be? Does the hippocampus or the limbic network become involved at some point And is there abnormal synchrony between perilesional cortex and the hippocampus that may lead to some of these events? Turns out that this same model of FPI that we talked about earlier is also an established model of post-traumatic epilepsy. 50% of these rats develop seizures within 6 months after FPI. And there are very few studies on the circuitry changes or EEG changes after injury related to seizures. There are a couple of reports of, again, these pathological high-frequency oscillations in the several hundred hertz range that are occurring perilesionally in the first couple of weeks that may predict late seizures in rats. And all of these studies further have been done either with skull screws as electrodes or as single channel depth electrodes, but there have been no investigations using high density arrays that are able to look across the laminar inputs of cortical tissue.
Can you do that in rats yet?
Hmm?
Can you do that in rats?
Can you, the high density array? Yeah, so that's what I was showing you with the,
Awake.
yes. So the lab has started to do that awake, and we are able to do that. We can do it wirelessly, as it turns out. Yeah. Well, no.
I just don't know if you had any experience making that actually work.
Yes, we have experience making that work. There's a balance that you still haven't gotten around of there's a balance between getting across more of the laminar structure and being able to record more units. So it really, in a chronic scale, in order to capture many units, you have to be able to drive the electrode down. And so when you do that, your field structure changes. But that's not an insurmountable problem. You can deal with that, but it's not, it's not as straightforward as it might seem. So, but then the question is, to what degree do these FPI models model relevant aspects of TBI in humans? How adequately does it capture the human condition? TBI is highly heterogeneous. The mechanisms of injury are varied. The networks affected are different from patient to patient. We have no idea who will go on to have PTE, aside from a few clinical correlates, who will go on to have cognitive deficits. We don't know. There are very few studies that are beginning to ask, are there electrographic markers that are acutely that predict the development of PTE later or of cognitive impairment later? Is there evidence of abnormal synchrony between injured brain and the hippocampal network? If we identify a pathological process occurring between injured brain and the hippocampal network, can we intervene? That period that period after acute injury, in days to weeks, is there an opportunity to identify the initiation and ongoing pathological process that could then be intervened upon? For example, could DBS be used to acutely arrest epileptogenesis? Not necessarily seizures, but the arrest of a derangement of a network that then may lead to seizures. And what about cognitive problems? So, for example, how might this look? You might have a study where you implant a depth electrode in injured cortex in the neuro ICU, along with other intracranial monitoring like ICP monitoring, etc. And maybe we can put a depth electrode in the medial temporal lobe. In that situation, we can ask some simple questions. Can we capture more early seizures or epileptic activity than we can from scalp EEG? If so, where is it? And does treating those events prevent early seizures, or does it affect longer-term outcomes? We don't know the answer to that question, but we do know it's likely that we will capture more epileptiform or epileptic activity. And also in that scenario, we can then ask, is there abnormal synchrony between injured cortex and the medial temporal cortex? Are there other electrographic events other than seizures that could be predictive of future network changes and therefore chronic deficits? And so that's basically where I would leave things. That's sort of the direction that I am interested in going. I will say that there are There are other sequelae of TBI. For example, there's a good proportion of TBI patients that develop movement disorders. I think there's an opportunity there, at least on the clinical side, to start to identify these patients and ask questions about what are the right targets. Tremor is a main movement disorder after TBI, as is dystonia. And there are only very few published studies as to the efficacy of the usual targets in these conditions. So with that, thank you. I have to obviously thank a lot of people from the Wolf Lab and other folks back at the University of Pennsylvania. So I will, of course, take your questions.
Do you know if there's any anatomical data on spine density or morphology in the deep layers that might give an anatomical correlate and explanation for this absence of?
Yeah, so there's actually a couple of very recent papers out of John Pavlichak's group that demonstrated a reduction in initial segment spines in the pyramidal cells in CA1 after using the same model after FPI, and using I want to say using intracellular techniques showed that may be a loss of inhibitory input at that area. So yes, there is the beginnings of that kind of evidence.
And you can begin to conceive of pharmacological or other techniques to try to intervene in that, that anatomical, physiological correlation.
Yeah.
I would say, as far as the epilepsy side of things go, there's a little bit of a disconnect between the epilepsy in animal models like yours, which are hippocampal mediated, and what we really see. We see epilepsy in traumatic brain injury, but we don't see a lot of hippocampal mediated epilepsy. We see a lot of epilepsy related to those I guess you'd call them perilesional, or the other areas, the input areas of the brain.
The ironic thing is most animal models, the fluid percussion is actually a better model of cortical injury than it is hippocampal injury. Whereas look at the hippocampus underneath, but what's getting injured is what's being taken away. It's actually cortex.
Yes.
That's an opportunity for study as well. I mean, in rat, obviously they're so close to each other. It's both models.
Yeah, and you also, as you said, the difference between the rat and pig. You miss the rotational aspect in the rat also.
Yeah.
It's such a huge component.
I think for all of those exact reasons, these animal models can allow you to ask specific questions if you want to ask a mechanistic question. But they're not really. They may not be modeling what's really going on in humans, and so you therefore may not be able to generate the right questions from your animal models. And so I think it's important to start doing these things in the human population in a specific way.
It's kind of complex, but have you guys ever considered, a lot of people that do depth electrodes or, epilepsy monitoring with depth electrodes will have sort of a standard array that they put in. And a lot of it is individually tailored to the patient's suggested epileptic focus and all that sort of stuff. But if there's sort of just fishing for somewhere where seizures are coming where they don't know, they'll put in just a standard array and see if they can tease out where it's coming from based upon known cortical connections. Has anybody ever considered a clinical trial with post-traumatic brain injured humans, saying that, okay, we've got all these biform contusions or temporal contusions or whatever they are. If we put in a standard array amongst all of these patients, we can kind of see where the cortical connections are disrupted via some sort of standardized pattern. That's kind of interesting.
I think so. I think that is something that I've thought about, but I'm not sure we are there yet.
Yeah,
I'm not sure we know.
It's a curative process right now. It's not quite in the emergence.
We don't even know the relevance of those early seizures that happen in the ICU, going forward. So I think beginning with maybe something that's a little more modest to put one depth electrode in when you're putting in your EVD, for example, we can start to just see what that activity looks like. Another interesting area that other than epileptiform activity is in that's getting some recent attention is spreading cortical depolarization. And there are a number of new studies out suggesting that that is a major component of what's going on acutely, not just in TBI, but in subarachnoid hemorrhage and stroke, and that this spreading depolarization,
Polarization or depression?
depolarization, I think so. For a long from the reading that I've done so far, there used to be a distinction made between the two. It's thought they were supposed to be two different processes. The thought is that they may actually be two different aspects of the same thing. But there's some evidence that patients who acutely have episodes of spreading depolarization have poor outcomes in TBI as well as stroke. There's some, but we know very little about how those electrophysiological changes may be impacting a wider network. So I think that's another opportunity for investigation.